Hello? This is not a cartoon. Sorry. Let me just go on to the next slide manually. Okay, this is a cartoon of that process. So we're starting here with our population back in time. We're going to run this through to the present, and we're going to have an allele frequency from blue to orange. So we're going to say, for the sake of argument, it's at low frequency is blue. So this is what we're going to see. What we're going to do, and now we've got a bunch, and so I'm looking, remember this is genomes, so we're looking at every allele throughout the genome, but I've just focused on one allele. We've now witnessed, if you like, the actual evolution, the change in time of this allele. Now, that's fine, that's super cool, but there's a very real problem with this as a way, you might say, okay, that selection has driven that change from blue to orange. Not, sadly, so simple. Because there's another way to get from blue to orange, and that's through admixture, that's through population mixing. So the notion here is we've got two historical populations. We're studying population 1 through time, but there's population 2, which is fixed for the allele. It's orange over here, okay? And we run this forward, and they're still independent. But then there's a migratory event such that this population encounters that population, and for whatever reason, let's say, takes over, shreds that population. So we've now got exactly the same outcome, a transformation from blue to orange, but we haven't invoked selection. This is purely due to admixture. So that's a big issue. Can we deal with that? Again, we can. This is ancient DNA. This is a stunning result. This is a more recent paper, 2018, again out of David Reich's lab. What we have here is, this is in Britain. We've just got a whole bunch of genomes over time, starting, 6,000 years ago until, about 3,000 years ago. And each line is an individual, and they're arrayed chronologically, okay? Now, they're not perfectly well sampled. As you can see, we've got many more samples from this point, about 4,500 years ago to 4,000 years ago. So it's not a homogeneous sampling array. And again, this is just down to the availability of specimens. But what I want you to say, it's obvious. It's spectacular. This is Britain, and it's color-coded by, this is the Neolithic British type. So blue is sort of what you might call the ancient Britain genome, the signature, the genetic signature of that population. What you have here, and they're established archaeological records of a movement from north of the Caspian Sea, populations which were sophisticated in their use of horses, for example, that moved west. And then the question always was, okay, they moved west, they imported their technologies, but when we say they moved west, was that the technology just spreading? Was this, if you like, a meme? Or were the people actually replacing other people? And that's what ancient DNA tells you. Boy, was this a replacement. I mean, you've gone to blue to red pretty damn cleanly. This is not a beautiful, I'm here, I'm a migrant, let's marry and have children. It's not. I'm a migrant, I'm going to chop your head off, okay? But these are exactly the kind of transformations I'm talking about, which could obscure the history of selection because you're bringing in different allele frequencies from different populations. But we know what those allele frequencies are because we've got data from the red people, right? So we know, so if we look at modern British people, we know exactly what mix they are. There are still vestiges of blueness, and we know what the historic allele frequencies were in the blue genomes, but they're also 80% red, and we know exactly what it was, what allele frequencies that the reds imported into Britain at this great cataclysmic moment in the history of my people. So that allows us to do allows Matheson et al. To do a beautifully sophisticated analysis. Yes, they're tracking the change of allele frequencies through time, but they're doing this in a way that can control for the input of variation of incoming alleles from migrant of admixed populations. And the result is this. So this is not quite your canonical, it is a Manhattan plot, obviously. It's not your canonical Manhattan plot because it's not, say, trying to identify a gene which is involved with a particular trait. What this is, so we've got each dot is a marker. We've got human chromosome 1 all the way through to chromosome 22. This is a sort of level of significance, the red dotted line, so we can basically ignore everything below that. Everything above this, and it's a complex statistical argument, shows significant change over their, I think it's about a 7,000 or 8,000 year period in the, if you like, prehistory of Europe, shows a significant change having controlled for admixture in the input from incoming populations. So these are the subjects of positive selection in, this is recent, of course. We're only talking about the past 10,000 years max in modern humans. But it doesn't get cooler than this. Just look at this. People, these geologists look unimpressed, but never mind. And I won't do this as a canonical way. People talk, genomics is incredibly boring because you come up with a list of genes and then you talk about this gene might do this or it does that. We think it might. I'm not going to do that. I'm just going to draw your attention to two. One is almost proof of principle. This is a gene we absolutely expected to see here. What is it? It's the gene which accounts for lactase persistence. So, as you probably know, humans by default turn off the enzyme lactase, which is used to digest lactose, which is the major carbohydrate component of milk, mammalian milk, once they're weaned, so at 2 years, let's say. There's no point in producing this damn enzyme because you're not going to be encountering milk, unless you live in a population which has access to dairy products. Dairy products only became a thing with the agricultural revolution, what, say 10,000 years ago. And so there has been strong selection in favor of mutations that keep that lactase gene turned on throughout life. So, you know, we expected to see this, and bang, there it is, the strongest signal in this data set. The other class, which I think is really interesting, is genes involved in various ways. One was involved in is implicated in resistance to tuberculosis, disease resistance genes. We already, again, sort of know this from contemporary studies. We know that diseases like malaria, Lassa fever have had an enormous impact in terms of shaping patterns of genetic variation on contemporary populations. Well, the same thing has been happening back here. And remember, these populations were relatively recently out of Africa. So they're encountering whole new sets of microbes. They don't have to worry about malaria if they're living in Scandinavia, but they've got to worry about a whole bunch of different other things. So again, you might a priori have predicted that you would see a lot of evolution traits like that. So that's my final point, is the second way in which ancient DNA can tell us about positive selection. The final way is perhaps even cooler. This is ancient introgression. Introgression is when I acquire genes, genetic material from an exogenous source, from an unexpected source. And there's a beautifully documented case in Tibetans. Tibetans, as you know, are fantastically adapted to living at crazy altitudes, at 3,000–4,000 meters above sea level. And people have done genetics to look into this, and they've fingered, and we've known for some time that this locus seems to be one of the key players in terms of how Tibetans manage with less oxygen than the rest of us, the EPS1 locus. Now, this is the cool thing. This is a comparison of the DNA sequence of this gene. And we've got a whole bunch of individuals. Each line here is an individual. And we've got two populations. We've got the non-Tibetan local people, i.e. or neighboring people, Han Chinese. And then we got Tibetans. Now, the gray is where everything's the same. So the black stuff, these are just DNA sequence differences, okay? And obviously there's a fair bit of noise down here, but notice that most the Chinese individuals here and some of the Tibetans have a sort of signature sequence. And the Tibetans have a different signature sequence, which really is very different. It's stunningly different. It's many base pairs different. It's a sort of quantum leap in terms of genetic differentiation away from the, quote, sequence. That's bizarre, because it's not like Tibetans are that distant genetically from Han Chinese, or from the rest of us, for that matter. But then comes, and this is the key thing, right at the top, there are two Denisovan sequences in here. The Denisovan sequences are the Tibetan sequences. So in other words, the key to Tibetan adaptation to high altitude is derived from that hybridization event, which occurred way back when with Tibetan ancestors and Denisovans. This has always been, so I mean, this is, look, there's no question, just because these alleles are so different between Tibetans and Han Chinese, and so similar between the Tibetans and Denisovans. There's no question this happened, but it still raises plenty of questions. The Denisova cave is not at high altitude. It's at about, I think, 600 meters above sea level. So why would the Denisovans have why would they have undergone natural selection in favor of high altitude adaptation? Well, as of 2 weeks ago, we now know. This is a paper that came out on the 1st of May. This is super cool. This is a new archaeological find in a cave in Tibet, high above sea level, of the first Denisovan material that we've identified not in the Denisova cave. So it's a mandible with a couple of teeth. If you want to watch it in action, there it is in glorious 3D surround sound. Unfortunately, it's about 160,000 years old. Unfortunately, there's no usable DNA, so DNA is not going to tell us anything. But isn't that cool? We sort of predicted that the Denisovans had to be living at high altitude, even though because somehow they were adapted to high altitude genetically and were somehow connected with the Tibetans. But we only had Denisovan material from Siberia at relatively low altitude. Bang, we found it at the second finding of Denisovan material is from high altitude. And this, by the way, looks rather lovely, doesn't it? These are Tibetan prayer flags. This was a site of pilgrimage, apparently, or is a site of pilgrimage where the find was made. So I just want to finish by going back sort of where I started. This is a paper I mentioned right at the beginning. This is Mary Claire King and Alan Wilson, 1975, made this extraordinary claim, extraordinary and accurate claim of 1% difference between human and chimpanzee DNA. And right then in that paper, they addressed the issue. It's kind of amazing. 1% difference, and yet we're pretty damn different, right? Anatomically, physiologically, behaviorally, cognitively. So how can you account for that little genetic change and all that rather substantial biological change? So now, how can we leverage a relatively small amount of genetic change to produce a lot of downstream phenotypic difference? And their solution was that it's, as I've already said, it's the control regions of genes. So they speculated that the key lay to the switches, the switching on and off of genes. I think they're right, and this is a cool final result that people have noticed that these human accelerated regions tend to be parked next door. Now, we don't know yet that those are regulatory regions, but the fact that they're HARs means they're functionally important. So this isn't in a gene, it's next door to a gene and is functionally important. That pretty much implies that it's involved in the regulation of that gene. And it's changed, and some of those, many of them, have changed rapidly along the human lineage. In other words, the more we go forward in this wonderful era of genomics and ancient DNA, the more this simple original speculation resonates. Thank you very much. Okay. Questions? No. A Neanderthal brain, and it depends exactly how you measure it, is actually very similar in size to our brain. And some people actually make the claim it's slightly larger. It's slightly difficult to measure because one thing that is clear, they're very different skull shapes. So we're very vertical. They have this big brow ridge and swept back. It's like they are pre-adapted for those Winter Olympic sports, like the luge, do you know what I mean? When they have those funny helmets. So no, they have big brains. So I would surmise, seeing as so Neanderthals have big brains, we have big brains, and Denisovans are the closest relative of Neanderthals, that they would be pretty big brained and therefore fairly smart. And this is one of the sort of revisionisms which is ongoing, and I'm not a paleoanthropologist, so I don't really know that much about it, but you know, there are fantastic B movies from the 50s which features the Neanderthal, and the Neanderthal is, you know, ravishing women and being sort of brutal and carries a big club. That's woefully, that's defaming the poor Neanderthals. So for example, those two mutations, two amino acid altering mutations in FOXP2 are present in Neanderthals as well. So there's plenty of evidence. For instance, they buried their dead with ceremonies and you know, with care. And you don't do that if you're, you know, just a random beast. So in other words, these were fairly cognitively sophisticated cultural beings. Then the question, of course, comes is how come we won and they didn't? And that I can't answer. I don't think anyone. I mean, people write endless screeds about it, but I don't think we know. Yes, sir. Yeah. Yep.